Morning, everybody. So, stream today is going to be again for general work help, and then answering any further questions and talking about science in the news if there are no questions. So, it's not going to be, super long, but just to give you a chance to have some help, no matter what you're working on. So on Friday, we started working on the greenhouse warming lab. I asked you to work on the guiding questions. If you didn't finish those on Friday, I'd be working on them now, finishing them now for tomorrow. If you're all kind of caught up on what you're working on and you have nothing to revise, then I would do some IXL practice, posted a link there, second in the agenda, to compare different types of thermal energy transfer. So the last IXL practice was about the direction of heat, going from high to low, using temperature to figure out where the energy is. This one's more of a comparison of where objects are in an environment and how it transfers energy in the environment. Conduction, convection, radiation, how those are the fundamental or how they're fundamentally tied to the laws of thermodynamics. And we're always interested in that first law, which is really the 0th law, because it was so important, discovered or finalized last out of the 4 laws, that these 3 types of heat transfer all exist because energy wants to reach equilibrium in our universe. Waste heat is that entropy. So when you're transferring energy, you're always going to have some waste heat. Okay? So again, you're finishing and making sure guiding questions 1 through 8 in the greenhouse warming lab are taken care of. Make sure you can go into Aspen, you can check your grades. DNA week 5 is recorded in there, as well as what's up. See how it impacted your average. See how you could maybe be happy with it or what to fix. And then the IXL comparison of different types of energy transfer. So you can ask your questions through Remind, obviously. And if there are no questions at different times, talk a little bit of science in the news. And I think the topic this time I want to focus on is the coronavirus. Our pandemic, COVID-19. Knowing the enemy is always a good thing. So how does this virus work? Some research done in South Korea and the United Kingdom has kind of come to light with its overall infection pattern. Johns Hopkins researchers have identified certain proteins that help it work really effectively, unfortunately, in our bodies. And then there's a young 8th grader down in Texas who actually identified a molecule that could kind of counteract how the virus binds to our cells. So an 8th grader, 14-year-old, doing a young scientist challenge across the entire United States. She found something that could maybe become a therapy or a treatment later on down the line. Because, this virus isn't going to go away, but it will hopefully evolve to something that is not as dangerous. Because viruses that kill its host are not good viruses in general of being a virus. Not just in general for us and our health, but they're just not good at what they do if they kill your host. Okay, think of like the flu, think of the common cold. Flu's a little more dangerous, obviously, but you get the idea. If it kills the host, it's not successful. All right. I got no questions so far. Chat, Remind, email, whatever you need. So I'm going to actually head over to University of Cambridge, which is in the United Kingdom. This right here is a look at what these researchers are going to be calling in this article, this little small published news release, a mini lung, all right? So these researchers, these scientists developed what they call a mini lung, so a model lung. We're still talking something microscopic, and they still tested this mini lung on a microscopic level, but just a little different than using a petri dish. And if you don't know what a petri dish is, it's literally a dish with some sugar on it that allow cells, bacteria, whatever you're trying to research to grow. Problem is, those dishes are flat, so they're two-dimensional. If you could build it in a dome, you can allow what you're studying to grow in 3 dimensions. And we know life exists in 3 dimensions, so you get a more accurate representation of how cells, bacteria, viruses work in a 3-dimensional environment as opposed to a flat 2D environment. Okay? So mini lungs were grown from tissue donated to Cambridge hospitals and provided a team of scientists from South Korea and the UK, again, Cambridge is in the UK, with important insights to how COVID-19 damages the lungs. Now, when we're talking COVID-19, that's the name of the disease. Hopefully you guys know that. Like, that's the name of the disease. I have the flu. I have the common cold. I have chicken pox. You know, when I was younger, I had chicken pox. You guys have a vaccine now. I have COVID-19. That's the name of the disease. Okay? Chickenpox is caused by a strain of the herpes simplex virus. Okay? Well, COVID-19 is caused by SARS-CoV-2. Maybe you've heard of this first term, SARS, severe acute respiratory syndrome. That was a pandemic that was actually around when I was in 8th grade. Came out of China, a coronavirus infecting people around the world, but was much less infectious compared to this current coronavirus. So this is the second one of these type of coronaviruses that cause major issues in the lungs, eventually developing pneumonia if it's not treated or if you're susceptible to these kinds of viruses, whatever kind of extenuating circumstances you have. So these researchers detail the mechanisms meaning how they interact with your body, underlying the virus and its infection and early immune response. Okay, so they kind of establish some data, 40 million cases of COVID-19 and then 1.13 million deaths worldwide. So this is a major issue. We know it's a pandemic. All right. And the main issue is patients developing pneumonia, whether that be because they're older, immune deficient in whatever way, certain conditions, certain diseases leaving that person. Okay, they can't have an immune response to develop to this virus. And they appear to attack the alveoli, hard to say that, which are the cells that exchange gases in your lungs. Okay, and that's what they're saying here. Take in the oxygen, exchange it with carbon dioxide. Okay, so we need to protect these cells, and they're deep down into your tissue of your lungs. So to better understand, they made mini organs grown in 3 dimensions, and I said that before. So again, you got to picture a petri dish as being flat, and then the cells can just kind of grow in colonies, whether they're bacteria or whether they're human or animal cells. They just kind of grow on a 2D plane. Okay, so they only have a length and a width, right? Well, these researchers kind of able to develop a kind of like a 3D petri dish. And so these cells can now develop extended tissue-like appearance, and they can get a very specific shape. Okay? And then that means now they have the 3 dimensions. They have length, width, and now they have height, right? And so we find that cells, they need those 3 dimensions to determine use of gravity and other cellular interactions, how they form tissue. So you don't really get tissue with option 1 right here. It's not as accurate as you would get in the lungs. This is a little more accurate and allows us to understand how a virus would maybe interact with actual lung tissue. So I'm kind of skipping over, the more, very more not scientific, but the very more detailed information about how they reprogram them back to stem cell So they took actual body cells, somatic cells, they turned them back into stem cells, which have the ability to really be a base cell, turn into any other cell. And then they could reprogram different cell types back to a lung tissue. But what they ended up doing is they introduced a strain from South Korea, a patient in South Korea, who was infected after January 26 from traveling from Wuhan, China. And they developed a timeline of the infection. And so they found out with the 3D models, when they were exposed to SARS-CoV-2, the virus began to replicate rapidly, reaching full cellular infection just 6 hours after infection. So the cells were pretty much all infected after 6 hours. Kind of zoom in. Okay. Just took 6 hours. That was it. And then around the same time, the cells began to produce interferons. So the cells themselves, the lung tissue, produce this protein, an interferon. Proteins act as warning signals to neighboring cells, telling them to activate their antiviral defenses. After 2 days, 48 hours, the interferons triggered the innate immune response, its first line of defense, and the cells started fighting back against infection. I did read a couple months ago that these interferons were actually being interfered with by the coronavirus, SARS-CoV-2. They were producing another kind of chemical protein, some factor that was binding to interferons and preventing the cells from communicating with each other. So think of like the interferon as almost like a flare being sent out. And that flare is going to get picked up by the other cells, and the immune cells, the white blood cells in the blood, are going to start mounting a response to that. So it took 2 hours for them to finally flare out to the immune system that, hey, there's trouble going on. And then 60 hours after infection, so we're looking at like almost 3 days, some of the alveolar cells began to disintegrate, leading to cell death and damage to the lung tissue. And that's what we want to avoid, right? That's where you're going to start having pneumonia, scarring tissue in the lungs. Although the research observed changes to the lung cells within 3 days of infection, so again, that 3 days, 60-hour mark, clinical symptoms of COVID-19 rarely occur so quickly and could sometimes take more than 10 days after exposure to appear. So that's why if you're ever exposed, or under quarantine, they tell you it's 14 days. You gotta give it that 2 week. So you give it a little extra time just to be safe. Why? Well, they think it takes time to really infiltrate the upper respiratory tract to get to the alveoli. And then it also requires a lot of cells to be infected. And so they're working with very small little model lungs. So it would happen a little faster. And so when you're working with a full person, it's going to take more time. But still, you're probably infected within those first 3 days. There's enough of the virus now replicating inside your body that it's going to take a hold. And then your immune system needs to respond to it. Then that's the issue. How does your immune system respond? Does it overreact, cause more damage? Does the virus suppress it? Do you have a weak immune system, and is it not going to be able to take care of the infection? Does it need a little more help, maybe in a hospital? Those are all questions we're worried about and what we want to keep studying. Okay? So this is just a simple timeline and understanding of how the virus infects. And if you're just coming in, if you're part of class, make sure your guiding questions for the greenhouse warming lab are done for tomorrow. Okay? If you're all set, caught up, no revisions, nothing to finish, you're not behind, there's an IXL link so you can practice understanding different types of thermal energy transfer: conduction, convection, radiation. Alright, nothing on our mind. Assuming you're all good. You guys are experts on the water cycle then. A little more advanced 5th grade version of the water cycle. So how about Johns Hopkins, one of our leading universities in the United States, especially in health. This picture right here is It said down here it's colorized. So this is a false color image just so you can see the differences a little better. But we do have a cell. I don't know if it's alveolar or not, or some kind of lung tissue, but it is a cell, human cell. And the little yellow things are the coronaviruses, SARS-CoV-2. So this just also shows you the size difference between the two. Between a virus particle, a viral particle, and the cell itself. A lot of the times you're talking virus or diagramming this stuff. You're in 6th grade or you go on to high school biology or college. It's still mind-boggling to me the size difference between a virus and a cell. And this is a larger virus as well. And there are some viruses that are actually almost the same size as cells. These megaviruses. So, viruses are really interesting. Were they a primordial form of life? Did they evolve after cells? Did they end up leading to cells? How did life really start? It's interesting how viruses might fit into that. Some scientists call them non-living. Some scientists call them an alternate form of life. They're kind of alien in how they interact or where they belong. Alright, so there is a lot of scientific jargon here. We're going to try and, like always, break it down, make it a little more accessible. Okay, but the main focus here is understanding that the virus has many ways of getting into the body, and it's going to focus on 2 main ways. Using a protein called factor D. Scientists have called factor D. A factor, to a biologist, is a protein. Okay, and then the ACE2 spike protein, right? That's what's going to give the virus its spiky appearance. That's how it's the main way it enters the cells. So this article this write-up kind of goes into how it gets into the cell, because that's what Anika here, that's what she developed, or what she researched a molecule for. To prevent binding of these proteins on the virus to your cells. Okay, and that's what other scientists are doing right now, too. So while the world waits eagerly for a safe and effective vaccine to prevent infections from severe acute respiratory syndrome coronavirus 2, SARS-CoV-2, the virus behind the COVID-19 pandemic, researchers are also focusing on better understanding how SARS-CoV-2 attacks the body in the search for other means of stopping its devastating impact. The key to one possibility, blocking a protein that enables the virus to turn the immune system against healthy cells, has been identified in a recent study by a team of Johns Hopkins medical researchers. So based on their findings, researchers believe the inhibiting the protein known as factor D, this is from the virus, because we're going to talk about a later factor in a second about the human body, factor H, also curtail the potential deadly inflammatory reactions that many patients have to the virus. So inflammation is your body's response to any kind of issue. We're talking, damage by either something physical, like, hey, I sprained my wrist, or I broke my ankle, or I got a bee sting, or I have a virus, or I have a bacterial infection. Inflammation is your body's response to any kind of damage or danger to the body cells. And so inflammation is primarily blood rushing to that area, swelling, and allowing white blood cells and other nutrients in the blood to rush to that area. Okay, it's like a first responder scene. Okay, so we need inflammation to move materials in our body. However, it can also be damaging because the immune system could over-respond, be dangerous to our own body, or it could cause swelling and damage into other locations in the body. So inflammation isn't always good, all right? And so what's really dangerous in terms of the younger patients that receive the infection that had developed COVID-19 is this inflammation. It's very rare, but still, it's dangerous in how this inflammation can kind of cause this overreaction to the virus. It's like, I can't get rid of the virus, so I'm just going to nuke all the lung tissue. That's very, very dangerous. Okay? So Scientists already know that spike proteins on the surface of SARS-CoV-2 make the pathogen look like, a medieval mace. That's their analogy. Corona, meaning crown in Latin, gives it a crown-like appearance. Are the means which it attaches to cells targeted for infection. So, our lovely little friend, I'm not going to use green. Yes, I want to keep sharing. So our coronavirus is spherical, and then on the surface, you get these little spikes sticking out. And you've seen, hopefully, diagrams. You kind of understand what I'm drawing here. It's tough to do it, freehand, but the purple there would be the spike protein. And the spike protein, when it's near your human cell, there's different things on the surface of your phospholipid bilayer, your cellular membranes. There's different proteins and sugars embedded inside the membrane because cells need to communicate, and this is how they're going to communicate. So, kind of want to draw this draw it like that, and we'll say the spike protein. It's kind of like this. Spike protein has a certain shape, and that certain shape, excuse me, got a tickle. That certain shape is going to bind to the spike protein in the coronavirus. But in addition, the spikes first grab hold of heparin sulfate, a large complex sugar molecule found on the surface of the cells in the lungs. So, I am going to go grab a drink of water. I will be right back. Alright. I swear it wasn't because of SARS-CoV-2. Okay? So, spike protein is going to bind to certain parts in the cellular membrane. However, kind of go back a little bit, it also binds to something called heparin sulfate, which is a large sugar that's in the membrane. So, in addition to that little blue module that we're going to identify in a second, there's also, I don't want to use that color, let's use, blue again. There's a sugar molecule next to the binding site to the coronavirus, the spike protein. And the coronavirus uses this as almost a little way to dock. So it's going to first bind to heparin sulfate, a sugar in the membrane, and then that allows it to kind of identify where the angiotensin converting enzyme 2, or ACE2 protein, is in the membrane. So the spike protein binds to ACE2 in the membrane. But to get there, it has to bind to this one first, the sugar. Then it binds to the protein. Okay, so again, I kind of refer to this as a docking system. It docks in, and then it binds and connects into the cell membrane. It's like a key going into a lock once you found the right address. Okay? And so they found out that when SARS-CoV-2 ties up to heparin sulfate, it prevents factor H, and this is from the human body. Factor D helps tie to heparin sulfate. Factor H is in the body, and factor H uses that molecule to bind to cells, and factor H is to regulate chemical signals that trigger inflammation and keep the immune system from harming healthy cells. Okay, so we got to kind of go back a little bit. There's, again, a lot of information, trying to simplify it where I can, but this little tether that I drew, this is factor, I'm just going to write D. This is factor D. So this is a protein from the virus that lassos and connects to heparin sulfate. Okay? And this prevents H from trying to get in. And what does H do? H prevents white blood cells and other antibodies from marking this by accident as a dangerous cell. You know, your immune system is responsible for cleaning out cells infected with viruses. They're responsible for getting rid of cancer cells. They're responsible for a wide variety of things. So anything without this, ID badge, factor H, is going to be targeted by the immune system. So really, this is kind of what I'm getting at in the beginning when I said CoV-2 isn't really a good virus. It's very good at infecting. It's really good at circumventing, immune response, but it's also kind of bad in that in order to get that spike protein in a better position, it has to bind to another molecule. And when it does that, it prevents the body from recognizing it as a healthy cell. Because let's say if SARS-CoV-2, what causes COVID-19, could just bind right to the cell without using this heparin sulfate, unless I'm missing something, it would make it more efficient. Because the virus would enter into the cell and would start hijacking the machinery to make proteins for more cells. And it wouldn't be detected. It would go undetected in doing that because the human body cell would still be able to have factor H come in and still regulate and not be triggering the immune system. So when it triggers the immune system is when it causes major issues. Okay, the immune system can go into overdrive, or the immune system isn't strong enough to fight it off. If it goes into overdrive, causes major inflammation. That's the issue with younger patients. Or it doesn't cause enough of a response, and that's what you get with older patients. Okay. Previous research has suggested that along with tying up heparin sulfate, SARS-CoV-2 activates a cascading series of biological reactions. Okay? And that's where you get this alternate system of inflammation and cell destruction, which is misdirected by the immune system at healthy organs. And that's, a major issue. All right. So I just want to get into that. It goes on further about other alternate pathways, but I just wanted you to kind of get a basic understanding of this virus. Its timetable of like 3 days is when infection is pretty much locked in. Nothing you can really do. So then you have to seek either, have your own immune system fight it off, which is most of the time what happens, or if not, then seek a doctor, medical attention. Okay? But most of the time, your immune system can respond normally and effectively. But there are, again, some cases where it can't, and that's the issue. That's the danger, right? So I don't want to spread it because we don't know who has these lacking responses in the immune system, or overreaction, really. Okay? So if we could kind of, let's use, so it's good, so let's use green. If we could just kind of find something that would like fit in right here, and prevent the spike protein from even getting close or being able to like move into the ACE2 receptor right here, the protein ACE2, well then the spike protein's useless and the coronavirus can't enter the cell. This has to happen. Factor D doesn't have to happen. This has to happen to have infection. So that's where Nika comes in. She just won competition, the 3M Young Scientist Challenge across the country, a $25,000 prize for a discovery that could provide a potential therapy to COVID-19. She identified this molecule as being one that could fit into the spike protein, prevent it from binding to our cells. Now, it doesn't really go into whether this protein could be safe or it has to be injected in a certain manner, a certain way. You can't just take compounds and place them in your body, but it could be, again, like she says, the basis for developing an antiviral drug, antiviral medication. So she discovered a molecule that can bind to the spike protein of the SARS-CoV-2 virus. She started off by looking at how you could bind a similar molecule to the influenza virus, because all viruses work similarly. They have some kind of protein that allows them into the cell. So initially her goal was to use methods in silico to identify a lead compound that could bind to a protein of the influenza virus. After spending so much time researching about pandemics, viruses, and drug discovery, it was crazy to think that I was actually living through something like this, said Anika. Because of the immense severity of the COVID-19 pandemic and the drastic impact it had made on the world in such a short time, I had the help of my mentor, like a teacher, professor, maybe even, or someone outside of school, could have been a researcher, doctor, changed directions to target the SARS-CoV-2 virus. Okay? She was inspired to find potential cures after learning about the 1918 flu pandemic and finding out how many people die every year in the United States, despite annual vaccinations and anti-influenza drugs. So again, and she's a dancer in her free time, she goes on to say. Okay, so she develops a molecule that could bind to the spike protein and prevent infection, which is how a lot of the antivirals are working. So we want a vaccine, that's the end goal, but we also want therapies that can help treat people who are already infected. Vaccines prevent infection, but what do you do for people who already have COVID-19? You need to give them some kind of medication, antiviral. Okay, antibiotics are anti-life. They don't work against viruses. They work against bacteria. So we need an antiviral in order to combat a virus. This could be one such thing. Pretty cool that an 8th grader did it. And if you want links to any of this, do your own reading. Cambridge, Johns Hopkins, or about Anika and what she did. I'll link it in the description later on today. I've always wanted to have you guys do this, or have somebody submit, but there's a lot going on this year. She still did it though. So again, got any questions, you can send them my way, now, later. DNA Week 5, and What's Up, Otter and Aspen. Again, as a reminder, join in late. You can check that out, see what you did, revising it, fixing and resubmitting. Anything is available to you. Okay. How to revise a check or your sheet. Go check classroom under references. Take a look at that video, explain how to do that. Okay. Stream today, we'll end it soon since no one seems to have any issues. Okay, and then make sure guiding questions 1 through 8 are done for Greenhouse Warming Lab. Gian, how you doing? How's 9th grade going, buddy? I knew a couple of you were watching. I know I got a couple siblings. One of them said you were watching. I didn't have your sibling, Gian. So Gian, hi. Rainy, if you're watching, because you didn't say anything last time. Hi. Good, I'm glad. You guess? Well, if you're healthy, that's a good thing. Physical science is the same thing you did in 7th and 8th, right? It's just review. You got any questions, I'll help you out. Yeah, you got a question? We'll ask them. Okay. Bye, Gian. Have a good day. Thanks for stopping by. So the rest of you, make sure that's taken care of. Try some IXL practice. Start doing some IXL practice on, I guess the atmosphere at some point. But we gotta work on that this week in the DNAs. Okay? So other than that, there seems to be no other issues, no questions. I will see you later. Ask questions if you got them throughout the day. See you tomorrow, or see you on Wednesday, depending on your last name. See you later.